afternoon we're going to have some talks on some more basic research topics, and something that's becoming less of a research topic and more mainstream is using immunotherapy for cancer and mesothelioma in particular. Mesothelioma in the past has not been felt to be a particularly immunosensitive or immunogenic tumor, but we're finding more and more that there are a lot of tumors that are actually sensitive to immunotherapy that weren't previously thought to be so. We're very fortunate to have Joachim Aerts here from the Netherlands. He's associate professor and head of the pulmonary and thoracic oncology department there at Erasmus Medical Center in Rotterdam. And he's had a long interest in immunotherapy and particularly dendritic cell therapies for cancer, and he's very graciously accepted the invitation to come here and tell us what's been going on in immunotherapy and specifically dendritic cell therapy for mesothelioma. So, Dr. Aerts.
Well, thank you very much for this introduction, and thank you for inviting me, and also for this nice conference and this very smooth organization, which it has been until so far. And I'm going to talk about dendritic cell-based immunotherapy. And these are my disclosures. And of interest is to tell that we have, as Erasmus University, have a spin-off company to make this to forward this dendritic cell therapy. So that's why I also am involved in that. And as you already heard, there are exciting times now for immunotherapy. So, I mean, I look in PubMed, and this is a slide from about a month ago, but then you can see that there's almost 250 hits, which is at least a little bit low number. I mean, until very recently, there wasn't very much interest for immunotherapy in mesothelioma. And certainly, a lot of research has been done, also by us, but also by others. And the end of last year, there was 2 important papers came out who also have revived the interest for immunotherapy very much. And this is the slide from the science paper from Dr. Hassan, who was here last year, I think. So you probably have seen this, but it's just to show that you can get impressive responses due to a form of immunotherapy. And the other thing is, which was already showed this slide about the tremelimumab trial, which is the anti-CTLA-4 antibody, which is now used into this randomized phase 2 study, which they also are taken now into a randomized phase 3 study where they expand the number of patients. And I certainly want to echo on what is said before, that these are a non-selected patient population. So we also wrote a comment on that in Lancet Oncology based on their first papers, that you can expect these trials to be negative because of the non-selection of patients. And I'll come back to that a little bit later. Well, and that's a problem which we are facing with now because of the role of immunotherapy as established in melanoma, but also in lung cancer, kidney cancer, other cancers. Everybody's stumbling over each other now to start on immunotherapy. And there are a lot of pharmaceutical companies, investigators are all starting up now to do this kind of research. And the risk we are taking is that we don't do a proper look at our proper scientific background and just do all these studies. And that's something which I'm very much eager to say, that we have to stress that we have to do this from a scientific background and not just do immunotherapy. I mean, when you look at the last World Conference, there was 4 years ago there was a World Conference, which is biannual conference. 4 years ago there was, at the last day, at the last hour, there was a session about immunotherapy on lung cancer. And now it was on the first day, presidential symposium, and everybody's talking now. People who refuse to talk to me because they don't believe in immunotherapy are now chairing that kind of session. So, and I don't know what their background is, because it's not that simple, immunotherapy. And we have to imagine, it's a little bit strange to say that as we are, every human will get one time or probably more often get some kind of cancer in his body or a pre-malignant cell. And our immune system is able to recognize that cell and eliminate it. And this is the way it goes. So normally you have a cell which is becoming malignant, expresses different antigens from before, and these antigens are recognized by antigen-presenting cells, for instance, dendritic cells, and these dendritic cells are then migrate into the lymph node, and there they educate the T cells to become directed to the tumor. So to say, the T cells are the effector cells. So the foreign antigens are recognized, just like with an infection. They are antigen-presenting cells, they move into the lymph node, there they educate the T cells to become directed against the tumor, and then the tumor is eliminated. But one way or the other, the tumor is able to, let's say, to hide himself from the immune system, or to suppress the functioning of the immune system. And it can either be done, for instance, by shedding of its antigens. When it doesn't show that it's foreign or becoming malignant, it will not be recognized. But it can also be done by building a collagen bridge around it, so that the dendritic cells will not recognize the tumor. It can shed cytokines which preclude dendritic cells to enter the lymph node. It can have an effect on the interaction between dendritic cells and the T cells. And it can have an effect on the T cells when they are present directed against the tumor, that it prohibits its action. And all the exciting news where everybody's talking about this PD-1 blockade is only there. So that means that the other mechanisms are not involved in that. So the immune system can fail by either the shedding of the tumor antigens or by an immunosuppression. And the thing is that we thought before that a tumor is just a clump of malignant cells, but what we know now is it's more than that. It consists of all this kind of immunosuppressive cells and also stromal cells, which all seems to have a function in the immune system. And the tumor can make antigens, can spread cytokines, and these cytokines can attract all kind of immunosuppressive cells, like myeloid-derived suppressor cells, regulatory T cells, inhibit the function of these dendritic cells, and then by this immunosuppression, which is regulated by the tumor, the immune system will not be activated against the tumor anymore. So on the one hand, we have immune cells which eliminate the tumor, like cytotoxic T cells, like NK cells, and on the other hand, there are immune cells which lead to tumor progression, like regulatory T cells, myeloid-derived suppressor cells, and tumor-associated macrophages. And the thing, of course, what we want to do is we want to tip the balance that the immune system doesn't work in tumor progression, but in tumor regression. And how can we do that? Well, to make it as simple as possible, I think you have to decrease of immunosuppression is to decrease the number of these individual cell types. And on the other hand, to increase immune activation, you can increase the number of that individual cell type directed against a tumor. And is that a problem, these immunosuppressive cells? Well, this is from data which we have collected in lung cancer, but I have similar data in mesothelioma. What you see here is these are myeloid-derived suppressor cells, and this is the number of these cells. And these are stage of lung cancer, stage 4 and lower stages, so less advanced disease. And what you see is that in these advanced diseases, there's an enormous increase in the number of myeloid-derived suppressor cells, which prohibit the function of this immune system. And on the other hand, you can have this increase in immune activation, as I told you. And you can do that by either giving a peptide vaccine or a tumor cell vaccine. You can give monoclonal antibodies, like these PD-1 antibodies. You have biological response modifiers, which I'm not going to discuss. And the other thing is cellular immunotherapy, which I'll come back to later. And why do we think that cell-based therapy is the way to go? That's probably because the effect of the tumor is that large that our immune system cannot be activated in vivo without help in some patients. And that is to say that there are patients in whom the immune system can be activated by this PD-1 blockade, but in other patients it can't, and then you need the cell-based therapy. And it's something which we know also. I mean, you have impressive responses in melanoma patients with this PD-1 blockade, but it's only in a very small amount of these patients. And it doesn't, so I don't say it doesn't work, but you have to properly select the patients for which you can give it. And why did we choose dendritic cells? Well, these are a special antigen-presenting cells, which are present normally in low frequency in the blood and the tissue, and they are constantly surrounding the environment for foreign substances. They're extremely potent antigen-presenting cells, and they induce and regulate these immune responses. And these dendritic cells can be activated in vivo, but also differentiated ex vivo from monocytes. So what you can do with patients, you can do a leukapheresis where you take out only the monocytes, and you can educate them outside of the body to become directed against, well, let's say anything you want, and then re-inject them into the patient. And that ex vivo activation of these dendritic cells, that circumvents the immunosuppressive nature of the tumor. So what we did in the trial and also proposed to take forward is that you generate ex vivo dendritic cells, which are incubated with antigens of the tumor, and you inject them into the patient, and then we hope to get an increase in the number of cytotoxic T cells, which then are able to have an effect on the tumor. Well, the thing is, which antigens should you choose? And everybody was searching for a common antigen in mesothelioma, and we know there is no common antigen, so not every mesothelioma expresses the same antigen. But what more important is that we also know now that it's probably not the best way to go, to only target one antigen. So, for instance, vaccination with just a MUC1 antigen is probably not effective. You have to get a pluripotent, multiprotein targeting. And that's also one of the things we see with this PD-1 blockade. It works better in squamous cell carcinoma than in adenocarcinoma, and we know that in squamous cell carcinoma there are more mutations, so more antigens present on the cell for which the immune system can be directed against. So, because of that, we choose to load these dendritic cells with autologous tumor cell lysate. So what we did is we took from the patients pleural effusion or tumor tissue, which we lysate, and then incubated them with their own dendritic cells, and then re-injected into the patients. So this was the first study, which we published now already 3 years ago, where patients were treated with standard chemotherapy, and afterwards, as a maintenance therapy, were treated with this dendritic cell-based vaccine. And what we saw, these are a little bit small, I think, but what we saw was that patients do get a response. 4 out of, let's say 3 out of the 10 patients did get a response on this immunotherapy treatment, but more important is of course also survival, but then you can say, well, this is a highly selected patient population, which I absolutely agree, but we see survival rates which are between 11 and, well, this is now over 7 years. So survival was, well, relatively good, but again, this is a very selective population, of course. Most important thing was this, and this is where we looked at, we took from the blood of these patients these cytotoxic T cells again, so we isolated these cytotoxic T cells, and we incubated that with their own tumor material, which we have left. And then we looked whether these cytotoxic T cells were able to lysate these tumor cells. And what you can see here is that before the treatment which we gave, there was no cytotoxic T cell present in the blood of these patients. So as we heard this morning, Dr. Cameron takes out about 700 milligrams of tumor, and then you have almost a kilogram of tumor in your body, and you don't have any cytotoxic T cell against this tumor. So that means that this tumor is able to shut down the immune system. And we saw that after our 3rd vaccination, we saw an increase in 4 out of 6 patients which we were able to test this. We saw an increase in this lysis. So then there were cytotoxic T cells developed directed against the tumor. But then again, you can say, well, the problem is still that you have all these immunosuppressive cells present. And that's absolutely a problem with our therapy. And I'm just going to discuss these three major cell types. And the first one is tumor-associated macrophages, because we don't have any good way of suppressing them. So we know that in mesothelioma, tumor-associated macrophages are a major problem, and they have a lot of mechanisms where they lead to tumor progression. For instance, they secrete VEGF, they are able to form blood vessels, they lower the function of increase the function of regulatory T cells. So they have a lot of negative effects, while the M1 type macrophages is the one we need, because they are also able to make an immune response against the tumor. So that's an ongoing story. For myeloid-derived suppressor cells, we looked in our murine model. We have a murine model which we developed to test all these experiments. And the first thing you see is that this myeloid-derived suppressor cells are a problem. So this is a histological biopsy of a mesothelioma tumor. But what you see here, these are the tumor cells, and these are all myeloid-derived suppressor cells, which are at the rim of the tumor. So these cells, you can imagine that these cells, well, let's say, shed the tumor from our immune system to be activated. And when the immune system is activated, these myeloid-derived suppressor cells express prostacyclin at their border, and prostacyclin is known to also lower the function of the immune system. So again, a lot of these cells are at the rim of the tumor. And then celecoxib, which is a prostacyclin inhibitor, is known to also have an effect on these myeloid-derived suppressor cells. And what we did then is we looked in our murine model what is the effect of this prostacyclin inhibitor on the number of myeloid-derived suppressor cells. And I just want to stress that this is at time 0 when the numbers are low of these myeloid-derived suppressor cells. Then the mice are injected with these tumor cells. And then you can see after 22 days that in the untreated mice, there's a large increase in the number of myeloid-derived suppressor cells, while in the celecoxib treated, it's lower. It's still there, but it's lower. And we also see then that when you look in the histological figures again, you can see that in the treated mice, there's a huge decrease in the number of these myeloid-derived suppressor cells. And then looking at the survival curves, again, these are murine experiments where we treat the mice with either placebo or a control mice, and then treat them with celecoxib, and then also treat them with our dendritic cell therapy, which we also have in the mice, and then the combination of the two. And then you can see when you have the control group, the mice all die after 21 days, and in the celecoxib group, the survival is increased, but even more when you do the combination of this dendritic cell therapy with this prostacyclin inhibitor. So showing that you can increase the efficacy of your dendritic cell therapy by adding something which decreases a certain immune suppressive cell. A similar thing is about regulatory T cells. So this is also a murine tumor where you can see the red cells are regulatory T cells. So they're largely present in the tumor. And you can add cyclophosphamide. So cyclophosphamide is a known chemotherapeuticum, but in low dose, it can be used to selectively inhibit regulatory T cell function. And you can see here again, the untreated mice die after 42 days, but then the mice treated with the cyclophosphamide live longer, and when you do the combination again with dendritic cell therapy, you see that also our dendritic cell therapy is doing better in the mice treated with the combination of both. So that we took forward to a clinical trial where we treated patients with mesothelioma similar to our former trial, but then we added up the cyclophosphamide in low dose just to have an effect on the regulatory T cells. And this is not quite good visible, but you see again that there's a nice survival. And I don't have all the data in detail yet, but there were no adverse events from this therapy, so that's obviously quite important. And the other thing is that we saw that in these 10 patients, not in all patients, the regulatory T cells were increased. So it shows that not every patient is equal, which we know already, of course, but also the immune suppressive mechanism of every tumor is different. So there are patients in whom regulatory T cells play probably an important role, but in other patients it will be the tumor-associated macrophages. Again, so showing that you have to select your patients for every therapy. So what we did then is we saw that in the patients with elevated regulatory T cells, we saw that the numbers were decreased when you added the cyclophosphamide. But we, of course, in patients where the levels were already low, you didn't see any effect. So there's also, again, this was a trial designed also a couple of years ago where you can say you have to first look at which profile per patient and then determine which is the best way to go. But how to take this dendritic cell therapy forward? So one of the problems we had was, I told you, we loaded these dendritic cells with tumor lysate of the patient's own material. But that's a problem and also a limitation. We have to screen about 80 patients to get 10 patients to be included in the trial because of the problems you have with enough and of proper tumor tissue. And also then the immunosuppressive effect of mesothelioma, which I discussed already, but I will add one thing up to that, and that's the effect of the tumor size. So here we treated mice, again, with dendritic cell-based therapy, but then at different time points after injection of the tumor cells. So the mice were injected with tumor cells, and then at later days they were treated with this immunotherapy. So these are the control mice. So mice are injected at day 0, and then you can see they all die 10 days after they got the tumor. And when you give this dendritic cell therapy at day 1, after you injected them with the tumor cells, then we know that the tumor is still quite small, and you can see that immunotherapy gives a prolongation of survival, and some mice even more. But when you give it at day 3 or even at day 5, then the effect is much less. So that means that the larger the tumor, the less effective your dendritic cell-based immunotherapy is. So it shows again that the larger the tumor, the more immunosuppressive effect it has, the less effective your immunotherapy is. And that's also one of the things why I believe that, but then from another way around, that pleurectomy decortication will have a major role in getting this immunotherapy forward, because as was shown this morning also, this is the effective way of a reduction in tumor load, and then you can give this immunotherapy as an adjuvant treatment. So what we did for the availability of the tumor lysate, so we have a new way now developed to not be in absolute need of the patient's his own tumor material, but we now have an allogenic tumor cell lysate. And that's also the reason why it took us so long to get this further, because that had to be under patent, and now we have an orphan drug designation both from NDA, from the FDA and the EMA to get this forward. So we're now planning to start a phase 1 study after the summer by using this allogenic tumor cell lysate in patients. So what do I think is the way to go forward? I think we have to decrease the tumor size in mesothelioma. Well, we have heard this morning that chemotherapy is probably not the best way to go, because it can have an effect, but its effect is limited. So I think that debulking surgery will have a role in this. I also think we have to determine the predominant immunosuppressive cell and then took the target, and also the target then to treat the patient. So I think there's per patient there is a different immunosuppressive profile, and you have to first identify that and then select which agents you are going to use to treat the patients at optimum. And then, of course, there are patients in whom this immunosuppressive effect is quite low, and these patients will benefit, for instance, from anti-CTLA-4 or for PD-1 blockade, but in others it won't. And I think that in these patients, we have to activate the immune system by using this cell-based therapy. And with that, I'd like to thank you for your attention.